Hi, everyone. Um, in a little bit, I'll introduce Bob Mallison, who will be the MC for today's wonderful event. The Lustman Awards is really one of the very highlights of the year for me. I look forward to it every year. It just doesn't get any better in terms of the convergence of the opportunity to hear great science, but also the opportunity to revel in the emergence of young investigators for whom this is sometimes their first or very early, one of their early public presentations. And I want to thank the Lustman family who's making this possible, particularly Susan Katz, Jonathan Katz, and Emily Lustman, who are here from the family, but there are many others who have long connections to this department, and Bob will give that history in a minute. I mainly wanted to just update you on a couple of things. On next Friday, June 1st, is the celebration of 100 years of women in medicine at Yale, and that will be a wonderful celebration. Many people, or several people from the Department of Psychiatry are speaking in the celebration, and I encourage everybody to go. On the 6th of June, which is the following Wednesday, Sam Quinones will be speaking here, and his book, Dreamland, which is a remarkable book about the emergence of the current opiate crisis. He'll be talking about that book and the problems related to the emergence of opiate dependence. That should be fantastic, and that's co-sponsored by the Department of Psychiatry and the Poynter Fellowship at Yale. And then on Friday, the 8th, we have our graduation, and during the grand rounds on Friday the 8th, our grand round speaker selected by the graduating class is an internationally famous psychoanalyst and teacher, educator named Glenn Gabbard, which should be great. And then in the evening, 5 to 8 or so, at York Place. York Place is a new meeting center on York Street. I think it's 150 York Street. We'll have our graduation ceremony, and I hope everybody will be there for that. In other news, I just want to let you know, and I think I can say this without too much risk that it's going to fall through still, that we received now, it looks like a total of about $6 million in federal funding to purchase a magnetoencephalogram, which we believe will be sited in the PET Center at the VA. And you might be wondering what a magnetoencephalogram is and why we need one, if it's so expensive. So, and I would say that's a good question. But I think the good answer is that we have MRIs which give you good spatial resolution in the brain, but very poor temporal resolution. And we have EEG, which gives you very good temporal resolution on the order of milliseconds, but very poor spatial resolution. It's hard to know where the EEG signal is coming from the brain. And we have this terrible problem in psychiatry, which is, and I think our speakers are going to help us wrestle with this question, is how do we understand the brain when to understand the brain means understanding its electrical activity on the scale that it operates, at the resolution, at the spatial resolution that it operates. And the thought is that having the magnetoencephalogram, which enables us to look at electrical activity in the deep structures of the human brain, combined with the MRI and combined with EEG or other kinds of techniques, will give us the kind of triangulation that we're going to need to move research in the department forward and then to the next step. And the reason that we got funding is because because the VA and the Department of Defense believe that the problems of post-traumatic stress disorder and traumatic brain injury, which affects the connections in the brain, both of these illnesses in a way, need this kind of deep insight to get it. So we're very lucky. This will be the first time a MEG has been located in a VA center in the country, and we'll keep you posted. It's going to require about a year's worth of renovation to make the facility ready for it, but I think that's good news. So, without further ado, Dr. Bob Mallison.
Welcome, everybody, to the 2017 Seymour L. Lessman Awards for resident research. There's a lot of good news today. First, we have 4 spectacular presentations by this year's awardees that now bring the total number of awards to 120 since 1974. I think the other great news is we have, at least by RSVP count this week, 17 total prior Lustman awardees joining us today for the event. And we're delighted to have you back. After grand rounds, we'd actually like to, in addition to getting a photo of this year's winners, we'd also like to invite Lustman awardee alumni to join us for a photo as well, if you can stay. The other great news is it's a beautiful day outside. And so with 4 presentations by 4 trainees, 4 mentors, and the weather, the corollary good news is that my comments will be brief. Listen, we're here to remember the remarkable life and legacy of Seymour Lessman. He was a member of the armed services who served our country in the Army in World War II. He got his PhD from the University of Chicago and his MD from the University of Illinois in the physiology and behavior of newborn infants. He became a resident in both adult and child psychiatry here at Yale in 1955, joined the faculty in 1962, and as many of you know, maybe some of you don't, 3 years later was promoted to the rank of full professor in the department. Among his academic works that he's best known for was the landmark textbook in the field Beyond the Best Interests of the Child, which he co-authored with Albert Solnit, Anna Freud, and Joe Goldstein. There is a plaque outside the auditorium that if people have a chance to look at on the way out, bears the name of the 120 awards that have been given in Seymour Lessman's memory. Thanks in no small part to the generosity of the Lessman family and Lessman Family Foundation. As John alluded to, John Katz, Susan Katz, and Emily Lessman are here today. We could just take one moment to thank them for their contribution to what really is a wonderful department of health. So with no further ado, I'd like to move to our first place award winner this year, Alexander Block, and introduce his mentor, David Glahn. David couldn't be with us today, so I'm going to read some remarks from David. David said, "I'm delighted that Dr. Aaron Alexander Block received this year's Lessman Award. Aaron is an absolutely outstanding research resident with a bright future in academic psychiatry. Indeed, given his experience in research, exceptionally strong analytic training, and scientific productivity, I feel that Aaron is much more of an academic equal rather than a trainee. Just to give you a feeling for Aaron's intellectual curiosity and productivity, he has already published or co-published 26 peer-reviewed articles, 10 as first author, on topics ranging from normal and pathological brain development to X-chromosome-linked intellectual disability to post-traumatic stress disorder. The basic, clinical, and translational aspects of Aaron's research are strikingly original, and while he has experience investigating different areas of psychopathology, his work has at least 2 major trends. First, Aaron works to understand normal development and how alterations in maturational trajectories lead to mental illness. Second, Aaron is an expert in graph theoretical and network modeling, something I hope Aaron will help explain to me. And the application of this analytic technique to big data to derive novel insights into psychopathology. This form of computational psychiatry is still relatively underutilized and holds promise to transform our appreciation for the complexities of mental illness and its impact on brain structure and function on the one hand, and on how psychiatric disorders fit with other medical diseases on the other. Today, Aaron will present a paper that we recently published in Cerebral Cortex that explores the impact of genetic factors on neuroanatomy. While this topic might seem a bit obscure, we have begun to apply the lessons learned in this work to better understand psychotic disorders. So with no further ado, let me welcome this year's first prize Lustman Award winner, Aaron Alexander Block.
So thank you very much for coming. It's an honor to be here today. My talk is about a study I did with David Glahn and colleagues on the genetic determinants of the thickness of the cerebral cortex. This works kind of at the intersection of brain imaging, genetics, and network science. So I'll give some background in those three areas before talking about the study. The analysis was done in a healthy population, but the motivation and also where it's going have everything to do with the neurobiology of mental illness. And I'm going to use schizophrenia research as an example because that's mostly been my area of focus. MRI allows us to study the thickness of the cerebral cortex, which we usually just call cortical thickness. And many studies have shown widespread areas that tend to be thinner in people with schizophrenia, showing the potential importance of cortical thickness as a marker of the disease process. And there's support for the hypothesis that in schizophrenia, this thinning may result from a loss of synaptic connections, which make up a lot of volume of the gray matter in the cortex. And the presumption is this relates somehow to genetics. As we've known for decades from twin studies that schizophrenia is highly heritable. Monozygotic twins are much more likely than dizygotic twins to be convergent in terms of their disease status. And now there are also confirmed loci of genetic risk, although with a few exceptions, we don't know how risk genes might mediate the risk, and we certainly don't know how they might relate to changes in cortical thickness. But this may be partly because we don't know very much about the genetics of cortical thickness in general. We do know that rare mutations of large effect can result in remarkably profound phenotypes. And also, we know that cortical thickness, like schizophrenia, is highly heritable. A high proportion of the variability across the population is due to genetic factors. If theoretically, different chunks of cortex could have specific genes that mediate genetic influence. There could be a kind of brain homunculus in the genome. Right? These are the prefrontal genes, and these are the cingulate genes. But actually, there seems to be a lot of overlap in terms of the genetic factors that influence different brain areas. And we start asking about shared genetic factors between two traits, which are themselves heritable. This sort of term of art is genetic correlation. So in our study, we were asking what proportion of shared variability in the thickness of two cortical regions is itself due to shared genetic factors. Looking at genetic correlations like this, there's now thousands of potential relationships between different brain regions. And this is where networks come in. Network science is basically a way of analyzing complicated patterns like this. And a property of networks that's particularly interesting to me is modularity. This is when a network can be divided into modules with lots of internal connectivity and relatively less external connectivity. So in illustration, it's common to color them different colors. And the point is that there tend to be more connections within the modules and fewer connections between the modules. From a developmental perspective, what's neat is that you can imagine modules developing semi-independently, and then there being a kind of process at work to connect them afterwards. And this is actually how man-made computational systems tend to be constructed. And there's been a lot of work across imaging modalities suggesting that the brain network is modular in this sense. And in fact, some really interesting prior work from UCSD suggested specifically that genetic correlations in cortical thickness have this kind of modular network structure. Using twin data, they showed these nice spatially coherent modules. The corresponding parts of the left and right hemispheres tend to be in the same module, indicating a high genetic correlation between these areas, which we call contralateral homologs. This research also showed convergence between modules derived from cortical thickness and modules derived from surface area, which together make cortical volume. But maybe the most interesting thing that this work proposed was showing that the best evidence was for 12 modules. That's one of the questions when you do this kind of analysis. How many modules are there? You could call this a process of looking for the best spatial resolution. Are there a few big modules, or are there many smaller modules? I won't get into the math, but the silhouette plot here is a measure of the quality of the modules. And this clear peak at 12 that you see suggests that 12 is somehow the optimal number of modules in this network. So we set out to do a network analysis of genetic correlations in cortical thickness using Dr. Glan's data, and specifically to see if we could replicate and extend this prior work. These scans were from 2,000 people from large extended families. This kind of family structure with lots of variability in terms of how genetically related people are to one another is what allows you to make inferences about heritability and cortical and genetic correlations, sort of analogous to a twin study. So essentially, we get the cortical thickness of lots of different regions in all these people from their MRI scans. We find the correlations across people. So the figure shows this for two possible regions, X and Y. You can see that as X gets thicker, so does Y in general. All the squares in this plot represent different people and the thickness of region X and region Y. But what the information about the family structure allows us to do is to then quantify how much of this correlation is due to shared genetic factors as opposed to environmental factors, which again is what we call a genetic correlation. And then when we get these genetic correlations between every pair of brain regions, they all can be represented in this giant genetic correlation matrix. When we look for modules in this data, at a first pass we find something pretty similar to the prior work. So again, regions that are colored the same were found to be in the same module. And I think you can see some similarities as far as the divisions. But what about this idea of 12 modules? This aspect of the prior work didn't seem to be supported. This is a step back to look more at the raw data. So here, each little square in the matrix is the genetic correlation between the cortical thickness of two regions. And we're trying to group together regions that are strongly correlated with one another. This is called hierarchical clustering, represented by these graphs with thin lines at the top and left of the matrix. We call these trees because of the way that they branch. Finding modules is sort of like cutting this tree. So if we cut the tree closer to the bottom, we're saying we only care about very strong genetic correlations. Specifically on the right, you can see if you only include the 5 strongest correlations, you end up with about 10 modules overall. But then if we cut the tree closer to the top, we also factor in weaker genetic correlations and we get fewer larger modules. There doesn't really seem to be evidence for one particular or most favored group of modules. And this is the same silhouette plot that we were showing before. In our data to the right, we're not seeing the kind of peak that would indicate one more natural number of modules. We also found a lack of such a peak in an independent, publicly available data set called the Human Connectome Project. And that's a twin data set. We also looked at alternative methods to find a particular favored resolution. But everything seemed to indicate a multi-resolution structure, where modules exist simultaneously at different resolutions across the cortex. And in a way, this is disappointing, because it would be nice to say we found 12 modules in the brain and there are really 12 modules in the brain. But on the other hand, this result is maybe more interesting and also more plausible, suggesting a more diffuse influence of genes across the cortex. We also looked at brain imaging patterns of gene mRNA expression. Sorry, we also looked outside of brain imaging at patterns of gene mRNA expression. We used the Allen Brain Atlas. This contains gene expression data from about 20,000 genes in about 1,500 cortical samples. And if you look at the correlation in gene expression profiles between these samples, samples in areas of cortex that we previously assigned to the same module also had higher correlations in gene expression. So to the left, you can see these samples illustrated at one modular resolution. But to the right, you can see we also found that no matter how many modules we divide the cortex into, gene expression is more highly correlated within the modules than between the modules, again supporting the idea of a multi-resolution structure. So if there isn't one special set of modules, how do we think about the organization of the system? We propose an alternative that is a more anatomical hypothesis, in which genetic organization of cortical thickness obeys two very simple principles. First, there is decreasing correlation strength with increased anatomical distance between two regions. And second, there are particularly strong correlations between contralateral homologous regions. And these principles seem to hold true both for the organization of genetic correlations in cortical thickness, which is above, and also for the gene expression patterns in the cortex, which is below. On the left, you see the general relationship that correlation decreases with distance between two regions. And on the right, you see especially high correlations with contralateral homologs. Biologically, this seems plausible because contralateral homologs share developmental precursors, and also because of the physical dispersion of neurotrophic factors and other signaling molecules during brain development. But then the question is, how do these relationships with distance and homology explain the modularity results that we saw? To get at this question, we simulated networks that were basically random networks, except they were constrained so that they had the same average relationship with distance and the same homology as the real networks. And it turns out that these simulations predicted the same kind of multi-resolution structure that you see in the actual data, which supports the idea that these anatomical principles are important potentially for the organization of cortical thickness in healthy individuals. So if there really were simple modules with clear boundaries at a specific scale, the obvious thing to do would be to look for the genes that influence those modules. That process hasn't really worked out. And one reason might be that we need to capture the multi-resolution nature of this organization. We're particularly excited to take these ideas and study how they relate with genetic risk in clinical conditions. And prior to my residency, my research focused on fMRI networks in schizophrenia. And we found evidence of alterations in brain modularity, as well as alterations in how anatomical distance influences functional connectivity. And these alterations are intriguingly similar to the properties that seem to drive the genetic organization of cortical thickness. Going forward, we're using fMRI data to look at the genetics of brain functional networks in healthy individuals and in people with psychiatric illness. If we can understand the relationship between genetic risk and alterations in brain network phenotypes, it would be a critical step towards understanding the biology of psychiatric illness and ultimately towards improving treatment for our patients. So thank you very much. I'd be delighted to take questions if we had time. I wanted to particularly thank the Lustman family for their generosity, Dr. Gahn and Dr. Crystal, my prior mentors who did a lot of the prior work that I showed as though it were my own, the NRTP program directors, Bob and Chris, and all the faculty and supervisors at Yale who, some of whom I see in the audience, and maybe especially my co-residents and my family, and most especially my son Emil. who turned 6 months old 2 days ago. Since Dr. Crystal asked once, he's not named after Emil Kraepelin, but he's still a big fan of the Yale Psychiatry Department.
Great talk. If there's a finite number of modules, like if there's 12, then one can ask the question, how do those modules map onto older data, like Brodmann areas, like, neurological lesion studies, and, these older ideas of what parts of cortex do where. And you can ask how those things relate. How do you even think about that question if it's a multimodular structure with no clear, proper level at which to make those comparisons?
That's a great question. I mean, it's definitely more complicated, that's for sure. I think one thing is that just because there's a multi-resolution structure doesn't mean that every single possible boundary is as good as every other single possible boundary. So you could still have a multi-resolution structure that is more or less similar to a modular structure that has a specific spatial resolution. If those fall neatly within the modular, the sort of multi-resolution modular structure at all levels, or if they tend to violate the boundaries at all levels, then you could still test for that kind of overlap.
So it was a wonderful talk, and there's a lot to chew on, and I thank you for the clarification about Emil's. There's been a lot of work to try to understand the modularity of the genetic risk for schizophrenia, and so people have looked at, used a variety of techniques like pathway analyses to try to compartmentalize, if you will, the biology. Like, these are involved in synaptic connectivity, these are involved in glutamate transmission, these are involved in inflammation or immune function, things like that. And I'm curious of whether you have or are thinking about harnessing that modularity to shed more light from either a mechanistic or structural point of view on the kind of modularity you've been exploring in terms of functional structural connectivity.
Yeah, that's a really neat idea. And I think another way of putting that would be like, how do gene networks relate to brain networks, kind of, right? And that, I think, is a really important question. I think, like, hopefully combining those things is exactly where we're going. I've been talking with a postdoc in Dan Geschwin's lab about like how we could maybe start to integrate these ideas. Yeah. I guess actually he's an assistant professor now, but I think we're really excited about that possibility. There's I guess there's the chance that it's just like a semantic similarity, where we talk about modules in genes and we talk about modules in brains, and so we're going to try to connect them, and they might not actually work out biologically. But I think there's more to it than that, and I think there's really some hypotheses worth exploring.
Next, I'd like to introduce Mohini Ranganathan, mentor for Joe Palladino, or as we know him affectionately, JP. She'll provide an introduction.
Good morning, everyone. And Bob, you just stole my first line.
Sorry.
So it is truly a delight to be here and be able to introduce João Paulo Dicino, or JP, as we all call him. JP is a soon-to-graduate PGY4 resident, and he joined our group, the Schizophrenia Neuropharmacology Research Group, as a PG3. Our group has extensive experience studying cannabinoids in humans, and JP was particularly interested because of his interest in examining the effects of cannabis and cannabinoids in bipolar disorder. So, despite a very heavy clinical load in his PG3 year, he jumped right in and took the lead in developing a protocol to study THC, the active component of cannabis, in individuals with bipolar disorder. As you may imagine, this is not an easy study to get through IRB and get FDA approval and to convince people it's actually a good idea to do. And it's truly a testament to his diligence and his determination to study this phenomenon in bipolar disorder that we did get full IRB approval and we got FDA approval and we successfully collected preliminary data that JP has now included in his NARSAD application that is under review. So while all this regulatory stuff was going on and took most of his awake hours, he also jumped into ongoing studies in our lab and to develop expertise in conducting these studies in a safe and ethical manner, including examining the effects of cannabinoids in healthy individuals and other patient populations. And he'll be presenting some of those data today. JP is just amazing to work with. He is a fantastic addition to our team. He will be joining an addiction fellowship this July, but we hope to lure him back and continue this line of work. And this past year has, I think, really been fantastic for JP. He is the recipient of several travel awards. He recently received the CDI award, and now he's a recipient of the Lustman Award. So I do believe this is a recognition of your hard work thus far, but also a recognition of your amazing potential. This past year was also fantastic for JP on a personal level. So he just got married. In fact, he just returned from his wedding a couple of weeks ago. And he's going to graduate in a couple of weeks. So really, he's at the threshold of fantastic things, both professionally and personally. And with that, I'll invite him to come and tell you about his study.
Good morning, everyone, and thank you, Dr. Nathan, for the lovely introduction. Special thanks to the Lustman family for the generosity in acknowledging this work. I'm really excited to share with you today the results of this recently completed human laboratory study. So in 2002, this was the cover of Time magazine, and it asked, is America going to pot? And this is the cover 2018. And essentially, it reaches that conclusion. And it highlights the need to understand the mechanisms involved in the cannabinoid response. As you know, there's a huge variation in the way people respond to cannabinoids. And this is one of the main interests of our lab. What inspired this work was arguably the first pharmacogenetics study in the history of psychiatry. And it has a very long title, as you see here, but it's also known as the Dunedin study, named after the town in New Zealand where the study was conducted. And what this group did was very remarkable. They followed over 1,000 individuals from birth for over 20 years. And the main finding they report in this paper is that individuals with a specific genotype of COMT, or the COMT enzyme, if they used cannabis, they had a high risk of developing psychosis. So we got interested in that. That was an observational study, and we decided to test that in the lab. So do COMT genotypes influence the acute effects of cannabinoids? And we decided to use both genetic and pharmacological tools to answer that question. So I want to spend some time telling you what COMT does and how it relates to our study. So COMT is the enzyme that breaks down dopamine and other catecholamines as well. That's especially true in the prefrontal cortex, where the density of the dopamine transporter is much lower than in structures such as the striatum. So there's a specific single nucleotide polymorphism that leads to a substitution of valine alleles to methionine alleles, such that the homozygote individuals seen in red here have a fourfold reduction in the activity of this enzyme. So the valine homozygotes have high COMT activity, and therefore, because they break down dopamine very fast during the prefrontal cortex, they have low prefrontal dopamine. The functional relevance of that is that their prefrontal-dependent cognitive performance tends to be lower. Because of a feedback mechanism, their dopaminergic signaling, the striatum, may be higher. And the main finding of the Dunedin study was that they had a high risk of developing psychosis if they use cannabis. There's also some data suggesting they have a higher rate of having substance use disorders. On the opposite end of the spectrum, you have methionine homozygotes that, because of their low COMT activity, they have high prefrontal dopamine and a better prefrontal cortex-dependent cognitive performance. So I know there's a lot of information here. What I want you to remember is red, more dopamine, hot, and blue, less dopamine in the prefrontal cortex, higher risk of psychosis, maybe more poorer cognitive performance. And the heterozygote individuals in green are in between because these are codominant alleles. So the two questions we're trying to address with this study, the first one is, do COMT polymorphisms contribute to variation in the response to THC, the main psychoactive component of cannabis? And what if we inhibit COMT acutely by giving people a tocopone, a COMT inhibitor that's used to treat Parkinson's disease? What would that do to the cannabinoid response? So this was a double-blind, placebo-controlled crossover study. We included healthy individuals that were exposed to cannabis, but without cannabis use disorder. They were first genotyped, and they participated in two studies. The first study we're calling the genetic study because we looked at the influence of COMT polymorphisms on their response to THC. And the second study, we're calling it the pharmacological study because we looked at the influence of tocopone on the response to THC. And these were our outcome measures. Subjective effects of THC, so the rewarding effects, feeling high with a visual analog scale, psychosis-like effects of THC, measuring with the PANS, a commonly used measure of psychosis, and the cognitive battery to look at the cognitive effects of THC was the CANTAB. So all subjects who met inclusion and exclusion criteria were first genotyped, and after baseline assessments, they received what corresponds to about one standard 9 joint of cannabis. That's 0.05 milligrams of THC IV over 20 minutes. Then we collected their data and followed them until they were safely discharged to go home. That was the genetic study. In the pharmacological study, I want to draw your attention to the fact only homozygote individuals participated, only the individuals who were on opposite ends of the spectrum. That allows us to get a better signal. I also want to draw your attention to the fact that before receiving THC, they received COMT inhibition acutely, pharmacologically, with tocopone. So this was the pharmacological study. As you can see here, we had 74 individuals, and they did not differ in a number of clinical and demographic variables. Our first result, subjective feeling of high. So I want to orient you to the slide. On the left, we're going to have the results of the study looking at the effects of COMT polymorphisms, or the genetic study. On the right, the effects of tocopone. On the y-axis here, we have subjective feeling of high. And in blue, we have the Val homozygotes. In matte, we have the heterozygotes. And in red, the matte homozygotes. So blue, low prefrontal dopamine, high stratodopamine. And red, high prefrontal dopamine. As you can see here, placebo does not lead to a high, but THC does. And we found, and the Val individuals seem to be more vulnerable to these effects. We found a trend for an interaction between Val genotype and THC. And this is consistent with the fact that they have a higher rate of substance use disorders. In the pharmacological study, placebo does not lead to a high, nor does tocopone. THC does, but once you add tocopone, we didn't see a difference. So the effects of THC or THC plus tocopone were indistinguishable. Our second outcome was the psychosis-like effects of THC, which do happen in some individuals. So on the y-axis, we have change from baseline in PANS total score. Placebo does not lead to psychosis-like effects. THC leads to mild, transient, self-resolving psychosis-like effects that occur regardless of COMT polymorphism. So they did not influence the psychosis-like effects of THC here. Once we gave people tocopone, we had a similar finding where tocopone did not alter the psychosis-like effects of THC. And this consistent with growing data on the limited relevance of dopamine signaling for cannabinoid-induced psychosis. Our next two outcomes are cognitive outcomes. The first one is a measure of attention called rapid visual information processing task. And on the y-axis here, we have time in milliseconds. So what you see here is that THC leads to a slight worsening in attention performance. And here we did find an interaction between Val genotype and THC. So the Val individuals were more vulnerable to the effects of THC. Once we gave people tocopone, we did not see a difference. So the attention-inducing deficits of THC occurred regardless of tocopone administration. And our last outcome measure, measure of working memory, the spatial working memory within errors. On the y-axis, we have mean number of errors. So higher scores means worse working memory performance. And here, as expected, THC led to an increasing in errors, so a worsening in working memory performance that occurred regardless of COMT polymorphism. When we gave people tocopone, however, as you can see there in the last column, it counteracted the working memory deficits induced by THC. Remember, this is just one dose of tocopone. So we had a lot of findings. I want to spend some time summarizing them. We found a selective influence of COMT polymorphisms on cannabinoid-induced subjective feeling of high, which is consistent with what we know about the prevalent substance use disorders in VAL homozygotes. Is it because they have more rewarding effects? And also in the working memory and attention deficits, which is consistent with a lot of work on how dopamine levels in the prefrontal cortex modulate cognition. Tocopone reduced THC-induced working memory deficits. So a future direction is to test more prolonged doses of tocopone to counteract cannabinoid-induced dysfunction as cannabinoid use becomes more common. And the lack of impact of neither COMT polymorphisms or tocopone on the psychosis-like effects of THC is consistent with the skepticism that has occurred after the Dunedin study of COMT and of cannabinoid-induced, of the role of dopamine and cannabinoid-induced psychosis in general. I joined the lab as a PGY3, like Dr. Ragnathan mentioned, and it has been an amazing experience. I also had the privilege of working with Dr. D'Souza, both as a research mentor and as an attending the clinical trenches. And I cannot tell you how generous and kind and supportive they have been. As Dr. Ragnathan told you, I sought their mentorship with a very specific research question, and they helped me design several studies that I really consider to be a roadmap for my career. And for that, I'm really thankful. And I think all fellows in the lab can speak to this. So thank you very much. Again, thank you to the Lustman Fund for the generosity. We do team science. I want to acknowledge another mentor, Patrick Skosnik, Rajiv Radhakrishnan, Jose Cortes, who are co-authors in this paper. This work started with Savita Bhakta, who was awarded an RSAD several years ago. She's now at UCSD. Dr. Nelson, Dr. Pittenger, again, thank you for the advice, the feedback. As I told Dr. Maxson earlier, this R25 was a catalyzer for my research career. The Yale residency has been my home for the last 4 years, working with our participants. Last but not least, my family, who couldn't be here today, they will be here next week for the graduation. And my wonderful wife, Alicia Londonia, who also happens to be my chief resident, and my in-house peer reviewer. Thank you. We haven't seen any, but Dr. Ragnathan has done quite a bit of work on sex differences, and that's a good question. I mean, steroid hormones do influence the response to cannabinoids and the rewarding effects of substance in general. So.
You showed PANS, I think that was PANS total, correct? Did you look at the subscales?
In one of the prior papers, there was a definition of clinically significant psychosis that is greater, an increase in 3 points or more in the PANS positive, and we didn't see a difference there. So we analyzed the data in different ways, and our first hypothesis was that the VAL, based on the Dunedin study, the VAL homozygote would have a clinically significant increase in just PANS positive, just a PANS positive subscale, and we didn't see that. But that's a good question. It seems one possible interpretation of the data is that cannabinoid-induced psychosis may be a matter of sensitivity of postsynaptic dopamine receptors and not so much dopamine release. That's one possible interpretation.
So thanks very much. From past experience speaking with prior Lustman awardees, after the initial moment of elation, hearing that they are honored with this distinguished award, usually follows a moment of terror when they realize they have to present at ground rounds in front of all of you. And this year in particular, with 4 awardees, I compounded that terror by letting them know they could only talk for 10 to 15 minutes. It's so much harder to present a coherent, focused, and brief talk than it is to give a long talk, and they're doing a great job. So, at this point, I want to welcome Alex Kwan, the mentor of Al K, to give an introduction to Al's work.
So it is my pleasure to present to you Al K. Al did his MD PhD at the University of California in San Diego. He's currently a chief resident with the Neuroscience Research Training Program here, as well as for the Clinical Neuroscience Research Unit. Over the last 4 years at Yale, I have to say Al has been extremely productive in both the lab and the clinic. And for that, he has been given a number of awards, including recently a Research Colloquium Award from the American Psychiatric Association. He was also named an outstanding resident by the National Institute of Mental Health. So there is a common saying that the pupils are the windows to the mind. So I like that quote, and there's also a lot of scientific validity to that quote. Evidence has told us that the size of our pupil can change. For example, our pupil dilate when we go into a dark room, and our pupil also dilate when we pay attention. So while it's clear that the pupil can tell us something about our internal mental state, to what extent that this might be a useful indicator for the diagnosis and treatment of mental illnesses is less clear. So that's where I think Al's work will come in. So he'll tell you some of his experiment looking at pupil in mice and try to link them to exposure to stress. So we're extremely excited about these results, and for one reason, the pupil is also something that can be also easily measured in humans. So that's something that I think Al is one of his interests in his next position as a research fellow in the VA here. So please join me in welcoming Al K to the stage.
So thank you, Alex, and thank you especially to the Lessman family and to the department for all the support over the past few years. So as Alex said, I'm going to be talking about pupillary fluctuations and how they relate to brain state after stress. So people have known for a long time that hyperarousal is kind of a core feature of PTSD. And there's a lot of dimensions of that people have looked at: startle responses, galvanic skin response. And people know for a long time, as I said, this is a description in Shakespeare of a woman speaking to her husband who's come back from war, and she goes on. It's actually a beautiful passage. She describes really the DSM criteria for PTSD if you read it. And on the right, you see there's a sort of lore that there's a quote-unquote 1,000-yard stare in military veterans who've been through combat, and that's depicted in this painting from World War II. So there are fluctuations of the pupil, and as Alex said, if you're engaged in an effortful task, as you see sort of in the mental task part of this pupillary diameter fluctuation curve in the middle there, the pupil dilates, but you also notice something else, which is that when the person is not engaged with the mental task, there are large fluctuations of the pupil that are occurring spontaneously, and those are kind of quenched by mental effort. This has been called hippus, this is a Greek word that means horse, so because of the galloping kind of pattern of those spontaneous fluctuations, and basically been studied primarily in reference to ophthalmology, where people were a little bit interested in this, but overall not well studied until fairly recently, when in rodents people realized that you could really define the states of cortical activity really across the cortex just by looking at the pupil. So several labs, including two that were here at Yale, Jessica Cardin and David McCormick's lab, noticed that you could look at the firing of individual neurons and the pupil at the same time and just see that they were profoundly related. And particularly interesting to us was this paper from Andreas Tolius's lab, where they did imaging of the axons of the norepinephrine neurons that sort of project throughout the cortex. And what they found in this work is that every time the pupil dilates, just prior, there's a huge spike in these norepinephrine axons. And these events are happening about every few seconds, about every 5 seconds or so, which would be about 0.2 hertz. And I'll ask you to remember that because we'll kind of refer back to that slow frequency elsewhere in the talk. So this was interesting to us because the locus coeruleus is where these norepinephrine neurons live is a highly stress-sensitive region of the brain. It's been implicated in various psychiatric disorders. So we hypothesized that perhaps by looking at these slow fluctuations of the pupil in the resting state, we might see some signature of both difference in stress states as well as how that might relate to cortical arousal. So we have two groups of animals here on the left and the right, and we have 5-minute recordings of spontaneous oscillations of their pupils while they're kind of running on a treadmill. And what you can see is that a day after stress, the blue curves, which are the unstressed animals, are really dominated by this kind of slow undulating fluctuation. Whereas on the right, although there is still a little bit of that, there is considerably less variability and considerably less at that very slow frequency. We can quantify this and sort of break apart the amount of those fluctuations at very low frequencies, which are kind of shown at the bottom left there, and that's where we see a difference, but not at sort of higher frequencies. Consistent with this idea that the slow fluctuations of the pupillary diameter, which are induced by the locus coeruleus, are altered and reduced after stress. This plot on the right is primarily for aficionados, but basically what it shows you is that the orange curve shows you how separated we can make these two populations, stressed and unstressed, just on the basis of this very straightforward kind of first kind of analysis of the slow fluctuations of the pupil, and it does fairly well. So the internal model that we have of what's occurred here is that the locus coeruleus drives the dilations of the pupil, and also projects throughout the brain to coordinate cortical arousal fluctuations. When stress occurs, we think that the locus coeruleus activity, and this has been shown from previous work, is shifted into a less variable, kind of high, persistent state of firing, which we're seeing reflected in the pupil. So to better study that, we wanted to look in the prelimbic cortex at the same time as we measure these pupillary fluctuations. The prelimbic cortex in rodents is sort of ortholog of the human medial prefrontal cortex and is known to be involved in fear and anxiety behaviors. The way that we record neurons is that we put in a virus which expresses a calcium indicator that fluoresces every time there's a spike in these neurons. And then we can separate out, as you see on the far right, the activity of about 60 to 100 neurons that we record simultaneously. Yeah, there it is. So what you can see on the right is that there are about 50 neurons in this recording, and every time there's a pupillary dilation, you'll see a kind of coordinated pattern of activity in those neurons. And then we can ask how those individual neurons relate to the pupillary fluctuations and also how they relate to one another after stress. As you probably observed in the previous movie, the sort of average of all those neurons is quite tightly coupled to the pupil. So this has been shown in other cortical regions before, and we've shown it here in the prefrontal cortex, that the orange trace, which is the average of the 60 neurons that you see below, is really right on top of the pupils, and it's also dominated by this slow fluctuation at 0.1 or 0.2 hertz. And if you look at those individual neurons, you can also see that they're quite synchronous with one another and also with the pupil and the mean of the population. After stress, which is on the right here, what you can see is that slow undulating fluctuation is lost. And now still the average of all those neurons is quite coupled to the pupil, but it's less dominated by the slow fluctuation. And then at the level of individual neurons, you can see by eye that each of those rows being an individual neuron that we recorded simultaneously, that there is less kind of synchrony between those neurons. We can sort of quantify that. This is a cumulative plot of how correlated these neurons are to the pupil. They're all pretty correlated, but there is a substantial reduction after stress. we can also ask how the neurons are correlated to the activity of the other neurons that are simultaneously recorded. So up until this point, we just showed that these slow fluctuations in cortical arousal state are reduced after stress, and also that we can see that in the pupil. But here you can see that the coordination of these neurons to one another is reduced. So on the right is the increasing frequency that we're quantitating, and then there's a kind of correlation between the activity of individual neurons at that frequency and the average of that whole population, which is that orange trace on the left. And what you see is that just generally, the blue curve, which is unstressed, is much higher than the red curve. So the main effect is that across the board, there is a decorrelation of this network. But even if you normalize out that distance, you kind of remove that, what you can still see is that there, at that very low frequency at which we knew the locus coeruleus drives the pupillary fluctuations, there is still a more specific difference of that slow frequency. We also propose a sort of simple model that could be used to define periods of high and low arousal. So as you may remember from the earlier slide, every time the locus coeruleus fires intensely, the pupil kind of starts to go up. So we can hypothesize that there are probably a simple model would be that there are two states, high and low arousal. And we can define those kind of on a second-by-second basis just based on the pupil, and then see how neurons' activity correlates to that. And what we see is that across the population of neurons, this simple model does really well at distinguishing periods of high and low neural activity. So to summarize, we found that stress reduces these slow pupillary fluctuations. We found that the pupil reflects a change in cortical arousal. So this change in the pupillary fluctuations is not just due to something happening kind of in the periphery, but really is still a wonderful marker of the central arousal state. And then we also find that there is a kind of decorrelated network straight after stress, specifically at that low frequency. And one way to think about that is that we know that the locus coeruleus, which kind of drives these brain-wide fluctuations in arousal, has different modes of firing, which correspond to different levels of arousal in the wakeful state. So wakefulness is not just one state, it's multiple states. So at the bottom left, in very low arousal, you have low firing of these neurons and kind of low response to a tone, which is shown in the arrow. Then in the sort of intermediate state, where the animal is kind of aware and awake and so on, you have intermediate levels of basal firing, and then you have a nice response to a stimulus. And then in the tonic high arousal state, which is known to be induced by stress, you have a high tonic activity, but there's no real change in the firing of those neurons after stress. Less variability, which is perhaps what we're observing in cortical arousal dynamics and in the pupil. And one way to think about that is that as opposed to being in this kind of focused, task-engaged state, the animal may be in a kind of scanning or distractible state. So we think that the slow pupillary fluctuations have some potential as a potential biomarker of stress, and we're eager to collaborate with people to determine whether that's the case. And we're also interested in digging a little bit more into the mechanisms of how this works. So many people for me to thank. So Alex, foremost, it's been wonderful to be in his lab. It's been a great intellectual environment with a lot of really wonderful people who've really helped me get this project off the ground. Guido Rausch, who was a very talented Yale undergraduate who collected a lot of the data that I presented. Farhan also helped get the microscope set up working in this lab. And everyone else in the lab helped tremendously as well. And my twins, Dash and Theo, my wife Jennifer, my parents for helping in many ways as well. And special thanks to the Lessman family and all of the support I've received from our department and through our department from external funding sources.
I'm wondering if there's any meaningful way to connect this work with, say, fMRI or even magnetoencephalography
Yeah.
in veterans with post-traumatic
Yeah, so absolutely. So I've been talking to Chadi Abdullah here about his work looking at resting state fMRI and how we might record the people and see the relationship between those two. I do think that there's a lot of potential to both use the human neuroimaging methods to validate that this is actually true in people, as well as to connect the worlds of rodent neurophysiology, where we have a lot of information at a very small scale with the kind of subjective responses of people. So I think that first thing to do would be to find out, like, do these oscillations distinguish between people in stress states? But after that, I think you'd really want to know, like, whether these slow fluctuations in arousal are something that you can observe in people. So yeah, it's a great direction.
Does the low-frequency pupillary fluctuation in some way relate with a peripheral marker of, let's say, sympathovagal balance, like heart rate variability?
So I haven't measured it, so I can't say, but there are several studies in humans, kind of in sort of the computer science literature and some other places where people have done some recordings of pupillary fluctuations and heart rate variability, and they do find that there is a strong correlation. And I suspect that, yeah, acetylcholine also correlates to the slow fluctuations. But I think that I think it's quite likely that when people look at heart rate variability, for example, they're looking at a similar underlying process.
So could you comment on whether this pattern is seen both in response to acute stress and chronic, or just one or the other?
Right, so the way that we've operationalized this is you have this PTSD model in rodents, you look a day later and you see if it changes. We've also looked a little bit at the time course of it, and we're still kind of working that out. So I don't have any firm answer for you for now. We know that it persists at least for a day. Thank you.
My spouse has always been acutely sensitive to my paying attention closely to her or not, and I think I have some insight now into why she. I'd like to finish up. Thank you all again for coming today. Our last awardee may be familiar to you. He's a prior Lustman awardee, Rajiv, and I'd like his mentor, Cyril D'Souza, to come up and introduce him.
I'll be brief. As you know, Rajiv won this award back in 2014. It's been a pleasure to work with Rajiv. I first discovered Rajiv in 2010 when we were doing a study in India at the National Institute of Mental Health and Neurosciences, where Rajiv was a project officer, and it became clear to me that Rajiv was a keeper. And so I invited him here to the States, and he was a postdoc in my lab for a few years and then did his residency. He's just about to complete a fellowship in research and is going to stay on the faculty thanks to a YCCI award. And hopefully, Rajiv, you will stay with us. I don't want to put you in a spot, but anyway. So without further ado, I'd like Rajiv to come up and tell you about some of the work that he's done, which was funded by the Dana Foundation. This was awarded to Rajiv and our colleague Patrick Skosnick. So, Rajiv.
Thank you. Thank you, Dr. D'Souza, for that introduction. It's been a long time since the last award and since India, but thanks for reminding me about that. So this is a study which is truly a testament to the research support here at Yale, which began as a small grant from the D3 Fellowship, which helped us collect some pilot data. And as Dr. D'Souza said, we were then able, with Dr. Skorznik here, we were able to apply for more funding. And this is a study looking at PET imaging of synaptic density in schizophrenia. So one of the core neuropathological findings of schizophrenia is that there is reduced synaptic density. But most of this literature comes from post-mortem data, and this has never been verified in humans to date. So this is a slide of post-mortem data, and what you can see on the top row, or the top slide, is a healthy person, and the bottom two rows are people with schizophrenia. And what's apparent is the reduction in the number of synapses in patients with schizophrenia compared to healthy controls. Now, there is a protein, synaptic vesicular protein 2A, which is present on virtually every synapse. Let's see if the pointer works. So on the synaptic vesicle are these proteins called SV2, and these are uniquely conserved. They're present on virtually all active synapses. And the other interesting property is that they are limited in number to about 5. And that, in that sense, measuring SV2A can also help us estimate the total number of synapses. Now, UCBJ is a PET ligand which binds with high sensitivity and specificity to this protein. And it's actually an analog of the anticonvulsant levetiracetam that we've used. And SV2A, the specific signal from PET, can then be used to measure synaptic density. Another important point to make is that this ligand has very good test-retest variability, which is just about 3 to 5%. And this is from an earlier study done here at Yale. So the aim of our study was to measure global and regional synaptic density in male patients with schizophrenia compared to age and age-matched healthy controls using the PET ligand. We restricted the study to just males because the post-mortem literature suggested that there were gender differences. And we also sought to examine the relationship between synaptic density and disease characteristics such as age of onset of psychosis, duration of illness, and disease severity. So this was a cross-sectional study. Patients and healthy controls underwent MRI, which is a 3 Tesla structural MRI. PET imaging was on a high-resolution tomograph, which is a scanner with the highest sensitivity and resolution that's available here at Yale. We used VT as an outcome measure, and I'll come to that. The analysis included gray matter masking to take care of regional changes in gray matter between schizophrenia and healthy controls, and the analysis was voxel-wise using SPM. So the inclusion criteria was primarily healthy males and schizophrenia patients aged 18 to 55. There were no other Axis 1 diagnoses except for nicotine use disorder, and there was no drug use in the past 3 months. The assessments included the PANSS, which is a positive and negative syndrome scale. The cognitive assessment was on the COG state schizophrenia battery, and Dr. Skorznik had incorporated an EEG measure, which is an oddball paradigm. Now, this is the basic demographics, and I know it's a busy slide, but the diagnosis is of 2 groups, the healthy controls in schizophrenia, and on measures of age, body mass index, the total dose of radioactive injected mass dose, or the specific activity of radioactivity, there were no significant differences between these 2 groups. Now, the clinical characteristics of the patient with schizophrenia, the average age of onset was 21 years. The duration of illness was as long as 21 years, and age at first hospitalization was about 20 years. In terms of the total PANSS scores, this reflects about moderate level of severity, but also to note that the PANSS negative symptom scale is fairly high compared to others. Now, we also calculated a cumulative antipsychotic exposure based on CPC equivalents and years, and cigarette smoking and alcohol use as well. Now, this is a representative image of patients with schizophrenia versus healthy controls. And as you can see, the amount of radioactive uptake in patients with schizophrenia is a lot lower, at least visually, compared to healthy controls. Looking at group differences, now we also calculated a composite VT, which is a sum or an average of all the ROIs of the regions of interest that we have. And there is a significant difference in terms of, so the red being schizophrenia and the blue healthy controls. There were significant differences in the composite VT, regions of synaptic density in the amygdala, the fusiform gyrus, and I guess the global cortical ROI as well, which is a measure of the entire cortex taken together, and also the ventral striatum. So again, this is the same data shown as a table. I just want to point out that the change or the effect size of change is fairly in the large range for most of these regions: the amygdala, the fusiform gyrus, ventral striatum, the cortical ROI, and the composite VT. Now, we also use gray matter masking. This is to account for differences in asymmetry in gray matter. And here too, the significance persisted in both the amygdala, the fusiform gyrus, the ventral and the ventral striatum. And the percentage change was about 8% change that we could see in these regions in schizophrenia compared to healthy controls. Now, we're also interested in what does this mean and how does this relate to the symptom severity or other clinical characteristics. And so the global cortical VT, which is on the y-axis here, and this is the PANSS negative symptom score plotted on the x-axis, and we found a negative correlation between synaptic density as measured by VT and the PANSS negative symptom score. So the more, the lesser the synaptic VT, the synaptic density measure was, the greater was the PANSS negative symptom score. Now, the PANSS negative symptom also correlated with the change or the synaptic density as measured by VT in the fusiform gyrus, the thalamus, and also the composite VT. There were no correlations with cumulative antipsychotic exposure, but our measure was fairly crude and based on historical recall. Now, we also looked at age of onset of schizophrenia, and this seemed to correlate with the anterior cingulates, the synaptic density measure. So the earlier the onset of schizophrenia, the lower was the synaptic density in the anterior cingulate. Now, looking at cognitive test performance, the anterior cingulate seems to appear here too, where the synaptic density in the anterior cingulate correlated with the total intrusions on the RAVLT, which is a measure of verbal memory, total errors on one-card learning, which is a measure of working memory, and total errors on the Groton maze task, which is a measure of spatial working memory and executive function. We also looked at EEG measures using the oddball task, and this is a spectral analysis or time frequency analysis of the oddball to target, and what you can see on the y-axis is the frequency domain, on the x-axis is the time domain. The healthy controls in response to target, what you see is an increase in the theta activity, which is in response to the target. And this is absent in patients with schizophrenia. And we also found that the synaptic density in the anterior cingulate correlated with this evoked theta power in the oddball task, suggesting that it may be a measure of, it's maybe relevant to context updating, working memory, and allocation of attentional resources. So in summary, this is the first study to our knowledge to examine synaptic density in vivo in schizophrenia. Male schizophrenia subjects showed global reductions in synaptic density as measured by UCBJ-VT compared to age and gender match healthy controls. The greatest reductions were in the fusiform gyrus, the hippocampus, amygdala, ventral striatum, thalamus, and cortical gray matter. The magnitude of change in these regions was greater than the test-retest for the ligand, which is about 3 to 5%, except for that in the amygdala, where the test-retest is about 9%. And synaptic density correlated with the PANSS negative symptoms, age of onset of schizophrenia, and cognitive test performance, and the EEG measure on the oddball task. A few limitations to remember is that VT is a ratio of the equilibrium concentration of the radioligand in the tissue compared to that in plasma. And so it includes both free, non-specifically bound, and specifically bound tracer. BPND is a better measure and requires a reference region. Now, the centrum semi ovale is usually used as a reference region, but there are abnormalities or white matter abnormalities in schizophrenia which prevented us from using this as a measure of synaptic density. With that, I'd like to acknowledge the funding agencies, the D3 Fellowship, the Dana Foundation, and the Schizophrenia Research Fellowship. Of course, the Schizophrenia Research Group at Yale, with Cyril D'Souza and Patrick Skosnick and Mohini, and the Yale PET Center with Rich Carson, Mika, and Nabil. And thank you very much.
Very nice presentation. I just have a couple questions. One is, did you look at either alcohol use or tobacco use as a moderator factor in your analysis? And did you ask about parental substance use, whether it's alcohol or tobacco?
So we did not ask about parental substance use of alcohol or tobacco. And we did not separate the groups because we had just 3 subjects who'd used alcohol extensively, and we'd limited the amount of tobacco use as well. So we did not look at that, but that could be a postdoc.
What about cumulative lifetime exposure to antipsychotics?
We did look at that, and we did not see an effect. But I think I should preface that by saying that our measure was fairly crude. It was just based on historical timeline follow back and medical records. But in our analysis, we did not find a difference. And that seems to be consistent with at least postmortem analysis as well, that antipsychotics per se did not affect synaptic density. Yes.
So based on, go ahead. No, go ahead. I'm good. Go ahead. So based on your review of the literature, so what do you think synaptic loss, is it more like a cause of schizophrenia or an outcome of suffering from the disorder for a long time, especially because these brain regions are affected in multiple stress-related disorder, and you perhaps, if you're measuring gray matter deficits with different, other than SV2A, you will see similar reduction.
True. I think it's a chicken or egg question about what came first. But I think even being able to say that there is some change in synaptic density would make a difference, because now we have newer compounds that can help increase synaptic density or change synaptic density. But coming to the question of what came first, I guess we don't know if synaptic density leads to schizophrenia, or does schizophrenia lead to synaptic loss?
Thank you. Very lovely presentation. I'm going to ask a simple question that has no easy answer, but I want to hear what you think. So in histological studies of postmortem brains, there's evidence for decreased synaptic density, if you want to call it that way. In fMRI studies that are looking at the same problems through different glasses, there's some evidence of increased connectivity, those are the terms used in schizophrenia. And how does one reconcile this, or what would be the way, how could one reconcile this down the road?
Yeah, no, that's a good question. I guess the structural and functional implications of these change may be very different. What we know is that synapses are lost, and most synapses we expect are going to be excitatory synapses that are lost. There's also some evidence that in schizophrenia there is a decrease in the inhibitory or the GABAergic synapses. So those changes may again, much of the fMRI literature, there are regions where functional connectivity is decreased. There are some regions where it is increased. So I think it is the balance of excitatory and inhibitory and what's lost to which greater degree in which region that may determine these changes that we see.
And just to follow up on that, I would quibble a little bit with the characterization of the fMRI literature as pointing primarily towards an increase in functional connectivity in schizophrenia. I would say actually, if anything, there's evidence for a decrease in functional connectivity in schizophrenia.
Maybe one more question.
Just to follow up to Shadi's question, you showed a slide that showed a correlation with regard to age of onset of schizophrenia. Now, the participants were all between 18 and 55. I don't know if you had enough, but you could look at an interaction between age of onset and duration of illness. You know,
So we did see an effect with the age of onset, but we did not see an effect with duration of illness. So that's maybe some things to do with the trajectory, with treatment, with other factors, but we did not really see that with duration of illness, synaptic density.
a wide range in duration.
We had 20 years. And I think there's some early literature suggesting that the decline in at least cognitive test performance in schizophrenia is in the first 5 years, and then there is a plateauing. And it possibly could be, there may be something there that it's not a steady decline with time.
It was striking because there looked like just two groups, like before age 20 and after age 22, and it was really where the separation was.
Yeah.
Thanks very much. Thanks to all the presenters again. Thank you all for coming. Again, we invite all awardees this year and all alumni up for a photograph. Thanks.